let's talk about the next question, that is question number 3 on the screen. Explain the process of secondary growth in the stems of woody angiosperms with the help of schematic diagrams. What is its significance? So, for this question, first of all, the process, we have to explain the process of secondary growth in the stems of woody angiosperms. So as we know that the process of secondary growth in the woody angiosperms takes place with the help of two types of cambia, that is Cork Cambium and Vascular Cambium. So moving on to the explanation of this answer, we have to draw the schematic diagrams of this. So first of all, we are going to draw the Vascular Cambium here, Vascular Cambium, and here we are going to make schematic diagram of Cork Cambium. Alright, so let's start to draw schematic diagram for Vascular Cambium. So this is your epidermis layer, epidermal layer or epidermis layer. Then you have cortex region, and inside this you will see vascular bundles like this. Secondary growth always occurs in dicot plants only. Secondary growth is not the feature of monocot plants. We know that. So, secondary growth occurs in dicot plants, in angiospermic dicots, and we are talking about the process of secondary growth in angiospermic dicots in stems, right? So this process or this secondary growth in angiospermic stems takes place with the help of two types of cambia, Cork Cambia and Vascular Cambia, and we are writing or drawing the schematic diagram for Vascular Cambium here. So this is your epidermis layer. Let's label this epidermis. Then we have cortex region. So this is your cortex. Now these are your vascular bundles in which you will find the primary xylem and primary phloem. So on the outer region you are having primary phloem, primary, I'm writing the primary like this, primary phloem, and on the inner side this you are having primary xylem. Now you will see the rings between primary xylem and phloem in these bundles like this. There are rings present between primary xylem and phloem. These are cambium rings or Cork Cambium. So we'll label this as Cork Cambium. The innermost region here you can see is pith, is known as pith, and the region or the cells between all these vascular bundles you will call them as medullary rays. So these are medullary rays, medullary rays, right? So this is the schematic diagram of a dicot stem which is showing you vascular cambium or showing you the secondary growth in the angiospermic dicot stem via vascular cambium. Now let's draw another type of cambium, that is Cork Cambium. Now you will see Cork Cambium we are going to make. So this is your innermost circle. Bundles are present, then I'm talking about the secondary growth happened. I'm drawing the diagram or schematic diagram when the secondary growth is being taken place. So this is your circle having bundles, then this is your secondary xylem, then this is your phloem, then the secondary phloem on the outer side, secondary phloem on the outer side, secondary xylem on the inner side. Then you have a layer, protective layers of cork, which is known as phallogen, and the outermost protective layer you have is phallem. This is you are having phallem. right? Now, this is your phloem. Let's label these. This is phloem. This is phloem. Then the second layer we have is phallogen. Phallogen. This is the third layer is your secondary phloem. Secondary phloem. Then you have this as secondary xylem. Now you are going to make rings, cambial ring and medullary rays. So these are medullary rays like this. These are medullary rays. These are medullary rays. So let's write first of all this is as medullary rays, right? And this is your cambial ring. This is your cambial ring. Here we have is cambial ring. Cambial ring. Right, so we have drawn the schematic diagrams for Cork Cambium and Vascular Cambium as well. Now, we know that the process of secondary growth in the stem of woody angiosperms takes place with the help of two types of cambias, that is Vascular Cambium and Cork Cambium. We know that. So let's discuss how actually the secondary growth in dicot stem takes place. First of all, if we talk about with the help of Vascular Cambium, then we know that in this diagram you can clearly see that these are the cambial strips. These are the cambial strips which are present in between primary xylem and primary phloem. These strips in dicot stem during the process of secondary growth, which is present between primary phloem and primary xylem, is actually known as cambial strips, and the name of these cambial strips which is present between primary xylem and primary phloem is known as intrafascicular cambium, intrafascicular strips. Now, here you can see the middle portion or the cells which is present between the bundles are known as medullary rays. So these cells of medullary rays are going to form another strip between these bundles here. So these strips formed from the cells of medullary rays are going to form another kind of strip that is known as interfascicular strip. So new strip is going to form like this. New strip is going to form like this from the cells of medullary rays. And this interfascicular strip formed from the cells of medullary rays adjoining the intrafascicular strip which is present between the primary xylem and phloem. So this intermixing or adjoining of interfascicular strip and intrafascicular strip makes a new ring which is known as cambial ring. Now this cambium ring or cambial ring is going to form new cells on either sides, that is on the inner side and outer side. This cambial ring have bipolar division. This ring cuts off new cells on both the sides. The cells on the outer side here are going to convert into secondary phloem, and the cells present towards the pith are going to differentiate into secondary xylem. So this is how the secondary growth takes place in dicot stem with the help of Vascular Cambium. Now if we talk about the another type, that is we have Cork Cambium, then we know that Cork Cambium produces secondary growth and all this secondary growth tissues. Cork Cambium produces secondary growth tissues and all these tissues are known as peridome. The tissues formed from the Cork Cambium which is helpful in the secondary growth are collectively called as peridome. Now this Cork Cambium is also known as phallogen, is produced secondarily from the epidermal cells of living tissue. Right? Now what happens in Cork Cambium? In this Cork Cambium, the cambial strips are going to form cork on outer side which is known as phloem and inner cortex or secondary cortex on the inner side, which is known as phalloderm. So this is how the secondary growth in dicot stem takes place with the help of cork cambium. This cork cambium is going to produce secondary tissues in the stem with the help of forming of cork on the outer side and secondary cortex, which is also known as phalloderm, on the inner side. Right? Now, the outer side of this, the outer side of cork, which is also known as phallum. Phallum is also having lenticels or aerating pores on it. These lenticels are helpful in the gaseous exchange. So this is how the secondary growth in dicot stems takes place. Now let's write answer for this question. We are writing like this. Answer number 3. The process of secondary growth. The process of secondary growth takes place with the help of two types of cambia. The one we have is Cork Cambium and second one we have is Vascular Cambium. So first of all, let's write that is Vascular Cambium. First we are going to write Vascular Cambium. Now, Vascular Cambium. In Vascular Cambium we'll write like this. In woody stems, what we see, the strips present between the woody strips present between the primary xylem and phloem is known as intrafascicular ring or strip. Now, the cells of medullary rays, forms the interfascicular ring between the vascular bundles which adjoining the intrafascicular ring. With adjoining interfascicular rings formed from the medullary cells is adjoining the intrafascicular ring. This results into the formation. Of cambial ring. Now, after the formation of cambial ring, this divides the cells. This differentiates or cuts off the cells on either sides, right? So this cambial ring cuts off the new cells. That means it forms new cells on either sides. On the outer side, it forms the secondary phloem. And on the inner side, It forms secondary xylem. Secondary xylem. Remember that secondary xylem always produces a large amount as compared to secondary phloem. So we'll write like this: secondary xylem. Secondary xylem always produces in large amount as compared to secondary phloem. Secondary phloem. So this is about vascular cambium, or the secondary growth happens in dicot stem with the help of vascular cambium. Now we are left with the growth, secondary growth in dicot stem with the help of Cork cambium. Let's write that also. So let's write Cork cambium. Cork cambium. Now we know that this produces secondary growth tissues. Secondary growth tissues which are collectively called periderm. So this this Cork cambium produces secondary growth tissues, and these secondary growth tissues all together or collectively are known as periderm. We know that. Now this Cork cambium is also known as, phallogen, and it produces secondarily from the supraepidermal, that means the outermost epidermal layer. This Cork cambium produced from the outermost epidermal layer, epidermal cells of living tissue. Right? Now what happens in the secondary growth, or how this Cork cambium helps in the secondary growth in dicot stems? So it produces the cork or phallum, because this cork is also known as phallum on the outer side, and produces the secondary cortex, also known as phalloderm, on the inner side, right? Also on the outer side of this Cork cambium, that means on phallum, they're having aerating pores known as lenticels, which are helping them in gaseous exchange. So on phallum there is lenticels helping in gaseous exchange. So this is all about the secondary growth in dicot stems of angiosperms with the help of these two types of cambiums, that is vascular cambium and Cork cambium. Now in this question we are left with the significance of the secondary growth. So let's write the significance of the secondary growth. We are going to write significance. So the first one is that it replaces an old non-functional non-functional old tissues with active new tissues. With active new tissues, right? Second one is the plants who have secondary growth in them are living longer. They live longer as compared to other plants. So the plants having secondary growth live longer as compared to other plants who are not having secondary growth, right? Then, all right, we have this two significance. The third one we have is this commercial cork. Commercial cork is the product. The commercial cork we are getting from the market is actually the product of this secondary growth only. So this commercial cork is the product of secondary growth. Also the wood we get is also the product of the secondary growth. So this wood is an important product of secondary growth, right? So with this we have completed the whole answer of question number 3 with along with the secondary growth, how the secondary growth takes place in dicot stems of angiosperms along with its significance. So this is the complete solution of question number 3.